the first part, the inaugural Demon Network UK DRI Joint Seminar Series. We've got a couple of fantastic speakers today. We've got Professor Valentina Escott-Price and Dr. Timothy Rittman. I'm going to be introducing both of those speakers. But first of all, I'm going to give you a little bit of background about what we're doing and how the Demon Network and the UK DRI are partnering. So we've just secured an official partnership between these two organizations, and this is our first seminar together, obviously, so we're delighted to get going. For those in the DRI, you might not be familiar with the Deep Dementia Phenotyping Network or the DEMON Network. We now have over 600 members, including clinicians and data scientists, dementia researchers and industry partners. It was initially envisaged as a UK network to harness the power of data science and AI for dementia research and healthcare, but we quickly realized that the interest in collaborating in this way was global, and we now have members spanning 6 continents. So it's an amazing success given that we've only just launched in November. If you'd like to learn more and you'd like to get the newsletters and become a member, it's free to join. So just go to demondementia.com and there's information there. It only takes a couple of minutes to join, very straightforward. We've now got a very substantial infrastructure of regional leads like Tim, also theme leads, and we have things like a clinical advisory group and a public patient involvement group. But basically it's all set up as an infrastructure for people who want to innovate and people who want to do things differently. We work together on grants and papers and all of the normal academic stuff in lots of areas that are relevant to the DRI, so things like experimental models and imaging and biomarkers, but it also goes out to other areas, including methods and prevention and clinical practice. So as I say, if you're interested in joining the network and you're not already a member, then go to demondementia.com. So for today's inaugural seminar, we have 2 fantastic speakers, as I said. After the introduction, we'll kick off with a talk from Valentina. That will be about 20 minutes, and then we'll have 5 minutes for questions that relate to her talk. Then Tim's going to be talking to us, and we'll have another 5 minutes, and then hopefully you'll all get a chance to pitch in with burning questions. So if you'd like to add questions to the Q&A box, which I hope you can find on Zoom, then please do that, and I'll do my best to field questions to our speakers. So it's my pleasure, as I said, to introduce Professor Valentina Escott-Price. Valentina is a UK DRI group leader who's based at the University of Cardiff. She's an expert in big data. She's got lots of experience using large data sets and is really interested in risk genes and biological pathways. Valentina began by studying mathematics at St. Petersburg University in Russia and went on to get her PhD in statistics from Cardiff. And she's very collaborative in the way that she works. She's played a big role in discovering new hits for Alzheimer's disease on the genome. And she, as I say, she works at the UK DRI at Cardiff. I'm really interested to hear more about mathematical approaches to studying mechanisms of disease. So without further ado, I'll let Valentina dive in. As I say, we'll have about 20 minutes for Valentina's talk, and then don't forget to put your questions in the Q&A. So thank you. Over to you, Valentina.
Thank you. I'm sharing my screen now. Do you see my screen? And I'll put it in a full mode. Yeah, do you see it correctly?
Yes, that looks good, Valentina.
Okay, so I have called my presentation today From Genes to Treatment, but of course it's ambitious and that's to attract your attention. In fact, what we are trying to do, we are trying to find a needle in a haystack. So why I will try to explain what it means and how we are trying to attempt doing that. So first of all, it's yes, I do work in the Dementia Research Institute, and of course, dementia, it's a very broad definition, which includes a number of different disorders: Alzheimer's disease, Parkinson's disease, vascular dementia, etc., etc. So although Alzheimer's disease is contributing 60% in average to the dementia cases, the other diseases, of course, should not be disregarded, and we need to look at them in a similar manner, but of course, treating every disease and disorder separately. Today, I will be talking mostly about Alzheimer's disease. Two reasons: not only because it's the most common, but also because we have good data sets, large data sets already collected. And that's the first reason. And second reason is that, well, basically in Cardiff, you do have strong group studying genetics of dementia. So this why, of Alzheimer's disease, sorry. This is why I will be talking about Alzheimer's disease most of this presentation, but of course, all our approaches and methodologies can be applied to different type of dementia. And I'm very looking forward to be cross disorder rather than concentrating on Alzheimer's disease only. So let's talk about Alzheimer's disease first of all. We know that Alzheimer's disease pathology may begin 10 to 30 years before clinical symptoms can be observed. And genetics is a good start to study disease or to find people who are susceptible for this disease as early as possible. Yes, there are some biomarkers, and biomarkers improving every time, every day, whatever, but genetics does not change over time. Okay, mostly, let's say that, be careful here. So if we can, potentially we can predict Alzheimer's disease early enough and use those predictions to either advise to individuals to change their lifestyle, for example, or to keep an eye on them, basically. What I want, why genetics? So this figure we've been just published today, the paper is out in our review, and Anne-Rieke Sierkma did actually fantastic picture of representing heritability of Alzheimer's disease. If you see smaller than 1% of heritability is associated with a very early onset of Alzheimer's disease, and that's very highly penetrant, a couple of very highly penetrant genes. For late onset Alzheimer's disease, we know ApoE4. It's been known for a while, and it explains 5 to 9% of heritability. A number of genome-wide significant loss have been identified, and its heritability is identified by this genome-wide associated loss is lower than ApoE, simply because ApoE has a strong effect, statistically strong effect, whereas other genome-wide associated loss have smaller effect and require large data sets to be statistically proven to be statistically significant. What we also know that SNP-based heritability, looking at the whole genome, is estimated between 24 and 53%. So by ApoE locus and other genome-wide significant loss, we explain whatever 20, 30% of that. And obviously there are many variations in the genome, which is associated with common variation, and that is all other assessed SNPs. Big chunk of graph here, which contribute to the polygenic score, to the polygenic nature of the disease. I will stop on this in a bit more detail. There is other genetic variation which is not picked up by the genome-wide association studies, and that is likely to be and still proven to be associated with rare variants, interactions between SNPs, and other genetic variation, perhaps copy number variants if we can find them, which should contribute to explain the whole heritability, genetic heritability of Alzheimer's disease. And also, of course, environmental factors are important. Therefore, the research which we are trying to do in Cardiff, it looks at all factors together, genetic to start with, because it's, first of all, we have data on that, and look into environmental factors like lifestyles, populational background to explain, or geographical location, if you like, to explain this extra environmental variable. And of course, we also expect that it's not that easy, just additive effect from genetics and environmental effects. Potentially, it's lots of interaction there. This is why it's not only simple analysis of simple data, it's big analysis and of big data. So earlier findings before the DRI started, we have shown in our early publication that Alzheimer's disease is polygenic disorder. So we have taken a large data set available at that time of about 30,000 cases and controls as an independent study, and predicted our cases and control by a polygenic component, which is collecting genetic information of many thousands of SNPs taken together in our sample in Cardiff. Well, it's of course not only our, but sample with collaborations, Jared sample, and we have shown that accuracy of prediction of Alzheimer's disease in our sample using an independent data set as a training sample is up to 73–75%. What we've done since, and so that was at the beginning of DRI, what we've done since that our discovery data sets increased in size, so we expect that effect sizes are much better identified with a better accuracy and better represented. Also, we worked a lot at standardization of polygenic risk scores, because if you look at the beautiful pink and blue graphs at the bottom of the slide, so blue are controls, that's a distribution in controls, pink is a distribution in cases. You see that they're quite separate, and if they standardize the score within the sample, you can see values between -4 and 4, as you would expect, and you see a number of people at the extreme risk for Alzheimer's disease or extreme being protective against Alzheimer's disease relatively small. However, it is in our sample. However, it's not clear how it is as compared to the general population. So what we've done recently, in fact, has taken 1,000 genomes population. It's a European population genetics, and in fact, UK Biobank, which I don't show here, but we've done it as well. And we have looked at our sample. So it's exactly the same sample as on the left side as compared to the population. And you can see that our sample has much more cases as we expect as compared to the population, and much more controls. That makes our studies more comparable, and looking at the polygenic scores of people in a particular sample more comparable and between studies and against the population. So since then, you can see the genome-wide association graph, and that was just recently published in Alison's good Lancet of Neurology review paper. You see that the data sets are much larger. We have many more peaks or many more genome-wide associated loci, and we improve standardization of our polygenic risk scores. And now we can use it perhaps to design biological experiments or to choose people for clinical trials based upon genetics and based upon their genetic predisposition to Alzheimer's disease. So what we also tested, we looked in the Alzheimer's disease neuroimaging initiative data set. We tried to predict people with MCI, mild cognitive impairment. And on the graph, curve graph in the top right corner, you can see blue are controls in the CIDNey data set, red are cases. They are separated as you expect, and people with MCI is somewhat in between, as you would expect again. So it reassures us that our polygenic construction works. Of course, there are a number of methodologies since then being published and tested how to derive polygenic scores, because the first approach was simply take an independent loci and make very simple model. There are a number of software like LDAC and SBIRS and Precise and LDPRED. And so we are trying to compare all these methodologies and make sense out of all these methodologies in order to identify the best predictive methodology. And also, if we design our biological experiment based upon polygenic risk score, we need to be consistent across. So that is a work in progress. And this violin plot, which you can see, it shows you on the x-axis different methodologies, and on the y-axis, it's a polygenic risk scores derived with these different methodologies. The lines which is in between show that some individual can show very low polygenic risk score according to one methodology and high according to another. or sometimes they are consistent. So basically we are working on this consistency to find the best approach, and we are looking for collaborations and for smart people and from people from data science to actually make sense out of all that. Next, what I wanted to say, there are a number of disease models which we can test, and number of propositions in the field. Oligogenic model, which in fact takes only the most significant associated SNPs. Polygenic model, which was originally approach was published in Purcell et al. 2014 for schizophrenia, and we are trying to see if that is the fact in Alzheimer's disease. And omnigenic model. So as I said, oligogenic model takes on genome-wide significant hits, combines them together, and we are trying to find out if it's the best one. Polygenic model is the one which we have published in 2015, and a couple of studies replicated this approach when we take not only genome-wide significant hits, but also SNPs with much smaller effects, which included into the score. And the last model is omnigenic model, which perhaps is a compromise between the two. So omnigenic model suggests that there are core genes which we potentially see as genome-wide significant in different samples, in different, potentially in different populations or geographical origins, let's say. And there is a number of genes which controlling the expression of these core genes. And this is why in some studies you potentially can see the only core genes contributing. In some other studies you can see more genes, because perhaps the discovery and replication data sets are much more homogeneous than the other studies. So we're investigating that. Please again contribute and talk to us if you're interested in that. So the next point what I would like to address, and that's really work in progress, and you're perhaps the first one who sees these graphs, looking at our heritability. Let's try to explain more heritability and try to model rare variants, if you're into the polygenic score or into prediction score. So if you model a rare variant as a part of polygenic score, it's perhaps will be hidden, like this needle in the hay, in a stack of hay. So we need to be a bit smarter than that. So what we are working on, we are trying to derive a probability of a disease, not a score between -4 and 4, but a probability of a disease mathematically defined, and the probability is a value between 0 and 1. So on this graph, we have you have black and blue curves. So the blue curve, it's a probability of Alzheimer's disease looking at all the population, 65 plus, and the black one in general population, basically in those which the disease prevalence basically 2%. You can see probability differs, and obviously you have much higher probability if compare blue and black line, for example, if you are in a risk group or all the group 65 plus, which makes sense. But also the lower graph shows that if you look at ApoE, we can predict the probability of the disease not very well. Basically, it just goes up to whatever 10% if we only look at ApoE. If we include an ApoE and genome-wide association signals, it's a mathematically based derivations, I must say. So you can see increase, and the mostly steep curves, they correspond when we take polygenic component into account. Of course, it's estimates. Of course, it's based on real sample, which we see in our case. And these probabilities can be recalculated as we improve the polygenic risk score prediction or change the models. With the rare variants it's not that easy. So perhaps if you have a very highly penetrant mutation to start with, so your probability of a disease much higher than in general population. So perhaps we should model them separately. So on the first graph, again, you have the same blue and black curve, and the dashed curves are exactly the same as you've seen in the previous picture, but the reason up, almost parallel curves, it's probability of a disease if you have very highly penetrant mutation like APP. So if you are unlucky and you have that mutation, your probability is very high to start with. And it's again up to for us to investigate if the polygenic risk score still required to push you over the threshold earlier to have Alzheimer's disease or not. So that's first one is for early penetrant mutation, and we simulate, estimated it using the like APP gene, you know about that. The other one is SORL1. It's relatively high odds ratio. Again, rare mutation, well, rare variant. So if you add it as a polygenic risk score, you will not see much effect on the polygenic risk. But if you model it separately, that's a second top right graph, right? You can see that your probability changes earlier, and you can pick it up. TREM2 we try to do, I mean, of course, much lower effect size and less change in terms of probability of the disease. ApoE. ApoE is not that easy. ApoE is not rare, and therefore you need to model it. You should not treat it. It's high risk or high effect size, but you shouldn't treat it as a rare variant being independent from polygenic risk score. Here you just simply estimate different disease prevalence if you have E4 allele or if you don't, and then you estimate probability in that way. So that's what we are working on, trying to bring rare variation and explain that green dashed line in our circle to bring rare variance into play. Again, how we do it further? Can we actually do something smarter than that? Can we, in fact, work with machine learning approaches to account for interactions between SNPs and genes, or to find these hidden patterns? And that is the next step which we are trying to take to address the issue of misinheritability, to include rare variants, etc. And we are making small steps in that directions. I don't claim that I'm specialist in machine learning. We can apply what it is, but again, we are very much looking for collaborations for machine learning. So what we tried to do so far, we looked at linear support vector machines. We looked at radial basis function support vector machines, which can pick up linear or nonlinear patterns. We looked at random forest. We looked at HD-Boost, which is a variation of random forest. And we tried to look at neural networks in order to pick up or find this needle in the stack of hay. So what we are trying to do with that, we are fitting these models and we are comparing prediction of case control status or whatever prediction classification, comparing it with classical multivariate logistic regression, which is not always the best, because when you have too many variables, you tend to overfit your models, and with a polygenic risk score with another extreme, which just takes one variable to predict your risk. So that's what we are working on. What we've done so far, I think some of you might have seen this graph from my other presentations. So what we've done, we actually tried to see if with machine learning approaches and with which of them we can pick up interactions, genetic interactions. So you can see A, B, C, D, E, it's a model of interactions. So it's not linear interaction, it's only for two SNPs, very simple model. And the goal, the second column, that's what we want to see, how separate two classes. So for example, for model A, you want to see all people with higher risk identified in the green box, and in the purple box to identify people with lower risk. And then you just follow the graph and see how it works with logistic regression, with interactions, and with different support vector machines approaches. Perhaps the most illustrative is the C one, the C case. So you can see that radial basis function SVM picks up the structure very well. So again, in real data, you will have a mixture of these structures, and some approaches will work better, some will work worse, and then that's what we need, again, try to address and make our progress on. Important to have in mind that machine learning approaches, sometimes could be interpreted as a black box. So it takes the data, trains it, come up with a model, black box, and then you're trying to see if it replicates. Even if it replicates, that is great. However, sometimes interpretability is not that high. So we can't tell clinician, or if a bit of this variable and that variable and this and that is present, because that's a black box. So that's another thing which we need to do, to unpack that black box and try to, well, find a compromise in explaining what works, how it works, and etc. So linear, non-linear, all that needs to be sorted. What we've done recently, we just published a review on machine learning prediction approach in psychiatric disorders, and you can find all sorts of prediction accuracies. You can see it was done for schizophrenia, for example, but you can see you can go anything from 50%, which is chance, to 90%. And the problem is, so we actually addressed it very well in our review, if you're interested to see, it's a data leakage. what can happen that you, not intentionally, of course not, but you might not tune the machine learning approaches properly. First, and quite often, and that's again not researchers' fault, when you have training data set and your validation data set slightly overlapping. That's an issue for polygenic risk scores, because now what we are allowed to share with other researchers, it's only summary statistics, and then to extract overlapping sample from summary statistics is very difficult, and assess which sample actually overlap is also very difficult. So sometimes we need to either come up with approaches to resolve it, or, well, obviously or sometimes we even don't know. But it's not only that. With machine learning approaches, you need to tune, there are extra set of parameters, machine learning hyperparameters so-called. And if you tune parameters in your both data sets and then predict that using prediction discovery data sets and validate it using parameters derived from both of them, obviously you are overfitting your model. So again, we need to be careful here. Next, what we do, it's our general direction to finish it up. Genetics doesn't explain everything. We need to come up with smart approaches to find hidden patterns in our data. We need to find that needle in a haystack. And we need to bring biological expertise and biological knowledge to refine our research. And of course, this is why our Dementia Research Institute is basically invaluable for my type of research. And for that, what we need a good data sets. In this, what we do have in Cardiff, and we're trying to analyze, it's a SEO data bank, about 3 million people, and the project starts 1st of October, started yesterday. Okay, so I need to chase it. So we will be looking at phenotypic variables, clinical records of about 3 million people from Wales. You'll have access to that, and we'll try to link genotypes which we have in Cardiff to those people and look at the genetic and environmental factors taken together. Of course, we have access to UK Biobank. It's 500,000 people. And lots of genetics and other information available there. We also have very good data sets like European Prevention Alzheimer's Dementia Consortium, not that big, but the data set is very clean. It will give us precision. IGAP data set, International Genetic Alzheimer's Project. Part of this data, we have access to all raw data, genotypes, part not. GERD data sets, it's our local data sets in Cardiff. GRACE dataset, we recently got access to that data. Again, we will look into this. And publicly available data like RoseMap, ADNI datasets. DPUK is a fantastic platform, and we're exploring collaborations with them to get access to all Dementia UK platforms with or without genetics. And again, trying to improve our prediction models. And perhaps it's one of the final slides here. What is real challenges is data sharing. Yes, we do have technical challenges like data storage, data analysis, everything, but it's nothing as compared with the real challenges with application for data sharing, for going through that, to apply with all regulations, and that actually prevents us from making a good research and making good progress on developing algorithms in testing them. Right, finally, in conclusion, what we would like to do, we would like to have fantastic prediction models for identifying people who will develop Alzheimer's disease to start with, or any other type of dementia at a later stage when we made our mistakes and learned from them. So, but also, we don't only want to know who will develop Alzheimer's disease, we also know when, how, and why. So when, it means that we want to predict age of onset very accurately. How, we want to know how quick will be the rate of decline of those people, so we can perhaps intervene or modify them in time. And why, of course, it's a biological mechanisms which are driving us to different type of dementia, to different type of dementia progression, etc., etc., etc. So that's what we want to achieve. I finish here, and I'm not sure. Yes, exactly 25 minutes. I'm good.
Thank you, Valentina. I really enjoyed that talk. It's clearly an area where we can make real progress. I think the fact that you can replicate those hits and see them in different populations is exciting. But as you've outlined, there's much work to be done to explain that unexplained variant. I'm also really interested in the way in which the gene interactions could be modeled. So Yeah, lots of work to be done there. We are running slightly over, actually. So what I'm going to do is I'm going to introduce Tim now, and then we'll take some questions at the end. So it's my pleasure to introduce Dr. Tim Ritman. Tim is a senior clinical research associate at the University of Cambridge, and he's also a consultant neurologist in the Memory Clinic and Disorders of Movement and Cognition clinics at Addenbrooke's. He's leading the bioinformatics theme of the Cambridge Centre for Parkinson's plus Disorders. He's done a lot of work looking at neurodegenerative disease and neuroimaging and linking it in with other data modalities. So I think it's really interesting to see how that complements the genetic work that Valentina's outlined. He's the Eastern Regional Co-Lead for the DEMON Network, and he's also an advisor to the World Young Leaders in Dementia Steering Committee. So over to you, Tim. Really looking forward to your talk.
Great, thanks, David, and thanks so much for the invitation to talk here. And that was a fantastic talk from Valentina. I think it's always difficult when you give these web talks to get any feedback, but that was fantastic. I really enjoyed it. What I'm sort of interested in really is understanding how we can take some of the mechanisms underlying particularly tau-related neurodegenerative diseases to the level of the whole brain in humans. So at the molecular and genetic level, there's some fantastic work around looking at protein aggregation, neuroinflammation, other disease processes, and it's striking to me that those mechanisms affect particular parts of the brain. They're particularly susceptible, which leads to these very recognizable clinical syndromes, which we see in the clinic. So what I'm going to try and do today is sort of give you a snapshot of some of the ways in which I think neuroimaging is helping to understand what's going on with these molecular pathologies at the level of the whole brain. So we'll start looking at brain networks and how they're susceptible to disease and resilient to disease. We'll look at how we've started to link neuroimaging to pathology more directly. And at the end, I just got a couple of quick examples of how we're trying to translate some of these sort of fancy neuroimaging technologies for clinical use. So firstly, tau is a protein that is very important in cells. It binds to microtubules, and in neurodegenerative disease, it comes away from those microtubules, it gets phosphorylated, and it folds in these abnormal configurations. And Michel Godart has sort of led the way with colleagues from around the world to identify how tau folds abnormally in different diseases, as we see here. And he just published the last one, I think, the CBD, just earlier this year. So that's the sort of molecular side of things. I don't really have time to go into the really interesting molecular aspects of tau. But from a clinical point of view, one of the diseases that we see is called progressive supranuclear palsy, and this is a cognitive and a motor disorder, and it's associated with tau. And it's a nice disease to study because when you make a diagnosis in clinic, you can be 95% confident that underlying that is a PSP tauopathy. For example, if you make a diagnosis of a similar disorder, corticobasal degeneration, you can only be about 60% confident that that's a CBD tauopathy. So this is a really nice description of PSP, and just picks out some of the aspects. So these people are incredibly slow, and they have problems with their eyes. So they get this frontalis muscle overactivity, which gives them this stare, which is very characteristic. They have restricted eye movement. So in this description, he had no more motion than if his eyes have been connected to the back of his skull. And they have axial rigidity, which means they don't bend in the middle. So when they sit down in a chair, they tend to just sit down on block rather than bend in the middle. And that comes with a sort of frontal dysexecutive syndrome as well, that people are just a bit sort of bloody minded often. And this is a really nice description. Well, of course it is. It's from Charles Dickens on his tour with Wilkie Collins around the north of England, where they got up to all sorts of things, including meeting this chap. And I think I agree with Andrew Lana, who thought that this was probably one of the earliest descriptions of PSP, this very classic prototypical tauopathy. So how do we link that sort of molecular side of things with that sort of clinical description? Well, we need something that's going to give us a sort of halfway house, something that we could measure, which is in between those two extremes. And I think brain networks are a good example of that. These are a predictable set of linked brain regions that we can identify using neuroimaging techniques. The brain naturally cycles through these different networks. And in my case, I usually use functional MRI as the modality to try and identify these networks. So you can use EEG or MEG, and they give very similar results. Now, the interesting thing about these networks in the context of neurodegeneration is that they seem to be roadmaps through which the neurodegenerative pathologies progress, so that they act as sort of pathways of susceptibility for disease. And this work was first demonstrated by some collaborators we worked with at UCSF and in Bill Seeley's lab. And they demonstrated, for example, that Alzheimer's disease targets this network called the default mode network. And frontotemporal dementia targets another network called the salience network. So it's not completely clear what it is about these networks which make them susceptible to disease. So a few years ago now, I decided to try and have a look at this, and particularly to look at whether tau expression was relevant. So I looked at 72 people with progressive supranuclear palsy and a comparable group with Parkinson's disease. So in PSP, tau accumulates, and in Parkinson's disease, tau is a risk factor, particularly for cognitive change. And what we found was that the areas that lost functional connectivity, so had less shared activity within those brain regions, were the areas that were most connected to begin with. And those areas that were most connected were the ones that had the most tau expression, and that's mapped as the gene for tau. And that was using gene expression data from the Allen Brain Atlas. And we went on to link those changes in connection strength to verbal fluency, which is a key marker of executive cognitive dysfunction in both PSP and Parkinson's disease. So that's sort of trying to, just a sort of initial step to try and understand what it is about some of these brain networks and the regions underlying them that make these networks susceptible to disease. But there isn't a one-to-one relationship between changes in brain structure and changes in brain function. So this is from our local cohort of PSP who have this Richardson syndrome, which is a typical presentation of PSP, combined with the Prospect data set, which is a national cohort. So this is, I think, about 78 people in this group. Just for the sort of eagle-eyed people, this is using the Brain-SOME Atlas, so it doesn't include the brainstem and the cerebellum, where you might also expect some structural changes. But we can see at the top there that we get changes in the basal ganglia and changes in the medial frontal lobe around the motor regions, which is exactly what we'd expect. And this is using a sort of Bayesian approach, which looks at sort of effect size combined with precision for that sort of statistical cutoff. We look at the next row down, we can see the connectivity is in similar areas, maybe a little bit more diffuse. And then below that, we see much more diffuse areas of change when we look at a measure of network transfer within brain networks, which is called closeness centrality, which sort of taps into the sort of efficiency of these brain networks. It essentially represents a randomization of the brain networks. We can sort of think about it in that way. So we can see that even with this sort of pathology, which is fairly restricted to the brainstem and sort of motor regions, it produces this very widespread change in brain networks. So structure doesn't always, or function doesn't always follow structure. So we've talked about how these brain networks can be susceptible to disease. but they can also be resilient as well. So for this, we looked at the genetic frontotemporal initiative dataset, which takes people with genetic frontotemporal dementia and their relatives who either do or don't carry the gene. And this is work from John Rohrer, which showed that the people who carry the gene for frontotemporal dementia have brain volume loss 10 years before they develop symptoms. So we're interested in what's happening in this pre-symptomatic period. And one of the things which is happening is actually a maintenance of that efficient brain structure. So this efficiency measure, this closeness centrality, during this pre-symptomatic phase, despite the loss of brain volume, is staying stable until the point that people get symptoms, and then that falls off a cliff. So that's sort of showing us that during this pre-symptomatic phase, if we can support those efficient brain structures, we might be able to push back the onset of disease symptoms, which would be significant for these people. Another way that we've been looking at these brain networks more recently to try and relate them to the clinical syndrome is to look at dynamic connectivity. So this is work by David Whiteside, who's one of my clinical fellows. This isn't published yet. We're just preparing a paper, so hopefully it will be in a preprint soon and publication in not too distant future. And here we're looking at a different brain states which the brain is cycling through, and we can pick these out with by using hidden Markov models. And this is looking again at I think 27 people with PSP and controls, and the PSP subjects are spending longer in frontoparietal states, which are often sort of connected to executive networks. And this taps into the theory of cognitive control, that to have efficient, flexible thinking, you need to be cycling through networks rapidly, and your brain needs to be flexible in that sense, to be flexible cognitively. And our PSP patients are very slow, and they're spending longer in these, almost putting more effort into these networks which represent executive cognitive function. And if we look at the time that the proportion of time people spend in those networks, that correlates with the severity of the disease. And we were able to, the nice thing about this data is that we were able to look at this in our local PSP cohort and replicate the same findings in an independent data set in the national prospect cohort of people with PSP. We went on then to have a look at what might be driving these changes in terms of structure and other functional measures. And it seems to be that this loss of complexity, this loss of sort of cycling through the networks, is related to subcortical volume loss. So that loss of brain volume in the subcortical regions of PSP that we saw earlier seems to be driving this lack of cognitive flexibility through this dynamic connectivity. And that also relates to this randomization I talked about, this sort of loss of efficiency of information transfer measured by this closeness centrality and cluster coefficient. So those sort of studies, I hope, have shown you how sort of brain networks are susceptible to disease, provide some resilience to disease, and are a way to be able to link changes in brain structure through to clinical cognitive changes in syndromes. What I want to show you now is just a couple of quick ways that we're starting to use neuroimaging to look at the underlying pathology. Now, I would love to have shown you some of this data from our own cohort. We have a cohort of people with PSP who have been through imaging studies and have come to post-mortem. Unfortunately, COVID has slowed down that analysis, but our great friends over at UCSF, led by Salvatore Spina in this case from the pathology department, has taken people from their brain bank with PSP and corticobasal degeneration, so two tauopathies, and shown that the neuroimaging volume loss is related to the loss of neurons and tau accumulation. So this gives us some confidence that actually when we're looking at neuroimaging data, we are looking at something which is relevant to the underlying disease. Now, I think there's a lot more that we can do with this. We can look at other aspects of neuropathology. and we can look at other imaging modalities. So I think there's a lot, has an exciting future. We can also try and look more directly using neuroimaging with PET. And this is work by Maura Malpetti, who's one of our very talented PhD students. And she looked at two PET ligands, one that binds tau, that's AV1451, and the other that binds activated microglia, the TSPO receptor, which is PK11195 ligand. And she was able to show in this cohort of people with PSP that tau pathology and neuroinflammation co-localize. And this paper is just published last week, so I'd really recommend you go and read it, because it's some very nice work. And I think this sort of highlights the value of neuroimaging, that we know that we think that neuroinflammation is relevant from epidemiological studies, from molecular studies to various neurodegenerative diseases. But now here, using PET, we can see that it's relevant in vivo in the human sporadic disease. So I think this really gives us confidence to go forward and look at treatments for neuroinflammation for neurodegenerative diseases, and particularly tauopathies. So finally, I want to just show you a couple of quick examples of how we're trying to use some of these methodologies to translate them to the clinic. The first is using DTI measures, which is work done by Marta Correa, who's a physicist at the Cognition and Brain Sciences unit. And I think I'd just echo Valentina in saying that collaboration is really important. Marta is not from a dementia background at all. She's from a physics background, but was able to come and use this data and show something really relevant and useful in a clinical space. So, Marta took the aspects of the DTI measures and used an SVM to classify patients with tauopathies, with corticobasal degeneration and PSP, against people with Parkinson's disease. Now, the important thing here is to, I think echoing what Valentina said earlier, is that we have to do this in the right way. And one of the important steps in this analysis was to pick out the right features to put into the SVM. And we did this with a PCA. So you take the DTI measures, put them through a PCA to pick out what are the important factors. You feed those into the machine learning algorithm, and if you just feed in the raw ROI data, you get these sort of accuracies here, so 70–80%, and suddenly when you do the PCA, that's bumped up to the high 90s. So there's real value in being careful about what data you put into machine learning algorithm to get a good result. And again, these are sort of validated in independent data sets. Finally, we want to take some of these sort of fancy imaging and machine learning methods right into the clinic. So I've been working with Zoe Kurtzi and Joseph Giorgio, who published this nice paper on prognostic predictive models, which take people with mild cognitive impairment and predict their future cognitive decline. And we're looking really to take this to the memory clinic. So started a study just recently called Quantitative MRI in the NHS memory clinics, which was began a few years ago with Dennis Chan in Cambridge, who's moved on and sort of taken it on and sort of re-implemented that, where people who come through the memory clinic who need a clinical scan have come to the WIBIC, our imaging centre, have a 3T MRI, and then also have a DTI functional MRI and a high resolution T2. And we can use that sort of research quality data, if you like, for machine learning for diagnosis and prognosis. So we started in neurology clinics. We're also shortly recruiting people from psychiatry clinics. And just highlight, this is an NHR portfolio study. So there's 4 other sites around the country who hopefully will be at various stages of showing interest in recruiting people for this as well. And this is not just validating in another cohort. this is putting in place getting data out of the NHS, putting the user interfaces in place, and linking these to very clearly to clinical pathways. So it just shows, there's quite a lot of effort in translating these models for clinical use. So I hope I've been able to show you that, starting with brain networks, that they're susceptible and resilient to neurodegenerative disease. I think the neuroimaging can help us understand how tau-associated dementias, particularly in the in vivo disease, and I think that's where it's really key, and that we can start to translate these neuroimaging and AI methods for patient benefit, both now and in the future. So thank you here's a few people from the Cambridge Centre for Parkinson's Plus, and thanks particularly to all the people that I collaborate with in Cambridge and further afield, and for all the funders who have made this work possible.
Thank you very much, Tim. I really enjoyed your talk as well. I think it's great to see how some of the discovery science is starting to percolate through in clinical practice. It reminded me of some of Valentina's work, actually, with again the discovery science around the genetics, and thinking about how that might translate into improved prediction or clinical practice.
Yeah, I think it's a really exciting time that people actually are starting to talk about that and ask those kinds of questions. So I think that's really notable over the past few years.
Fantastic. Valentina, we had a question about the kind of modeling that you're doing, and I wonder if you could reflect on whether you've had any frustrations in terms of the data that you've been able to use. It seems that you've gone to huge amounts of effort to combine data, pool data, and squeeze everything that you can out of it. But you must be hoping for more. What's held you back, do you think, in that respect?
It's not only size, it's also data quality, of course. So it should be a good compromise. To get, we put together data sets, but they are quite often not too comparable. And that also takes time to sort them out and understand what's right, what's wrong, but that's okay, it's technical issues. What holds me back, it's access to good clinical, curated information. So genetic data we've got, we kind of know what to do with genetic data in our sample. What we can't do, when we model things in our sample, I can't go to other people's sample and test these models because I have no permission to use other people's data. So then they are using their methods, reading my papers, but always the devil is in the details, right? So what exactly we've done, what we've done right, what we've done wrong. So that is this lack of collaboration due to lack of data sharing. That's perhaps holds me back on genetic side, but I'm actually taking genetic predictions further into clinics. I don't have good clinical information for our data set. For some subset I do have, but I need larger data sets to model it. And in different data sets, different variables are present because different cohorts, like for example in DPUK, they were designed to pick up different effects. They were basically collected to answer different questions. So we need to, first of all, get permissions for all of that, but that's, I'm getting help from DPUK, thanks to them. But then put it together, unify it together, and we are not there yet. So basically we are still struggling on the previous permission step.
It seems like we can try and address some of those difficulties through the data analytic approach itself, trying to move models between different data types. But it seems like actually the thirst to collaborate and innovate is actually probably more important.
Yes, and also I think I have heard, but I never tried to use it. There are approaches when we don't need to physically move the data from one server to another, but there are some solutions to combine the data virtually wherever it is, and then analyze together and then have results. Again, I'm not expert in those technological solutions, so those would be great. That expertise would be absolutely great to have.
Well, I mean, it was great to hear. I mean, you're obviously open to collaborations,
Absolutely.
Valentina, and that's great to hear. So, Tim, thinking about the neuroimaging then, to what, I mean, it gives us a fantastic insight into what's going on, but is it a little bit fatalistic? Are we just looking at patterns of disease after the fact, or does it give us some kind of insight into what might be driving it?
Yeah, I think with neuroimaging, you have to, I think, as always, you start with the right hypothesis. And I think, if you have an idea that, so how the disease might start and how that might affect the neuroimaging patterns that you see, I think that's one way to look at it. The other way to look at it is with some of these genetic data sets like GEMFI and the DIAN study in Alzheimer's disease, which are looking at people who carry genes for these disorders. And then you can sort of try and get to that pre-symptomatic stage. And then you can start to look at early causation. People looking at mild cognitive impairment as well as a sort of pre-Alzheimer state, and even using biomarkers to try and get into that sort of very earliest stage as well, to this sort of concept of pre-diagnostic Alzheimer's disease, which I think is interesting. I mean, I think you can only ever say that they're risk states, but I think it is a good way to try and get the earliest diseases. And then you need longitudinal imaging studies, I think, to really see how those changes develop over time. So I think the imaging is reflecting the pathology. Yeah, it probably does lag a little bit behind, but I still think it's useful if you can see those changes over time and pick out those patterns, with it starting with a sort of strong hypothesis of what you expect to see.
Absolutely. I mean, I think the fact that you can see changes so early is incredibly promising, and obviously each of the different types of models have their own limitations, but I guess that's also an opportunity, isn't it, to start to build models that can transfer between different populations or between mouse models and humans and so on. There's a lot of scope for model building, isn't there?
Yeah, I think there is, and I think with the sort of advent of using machine learning and AI and so on to try and work out use those models to pick up some of the variability and differences, so that you can try and make those jumps between, as you say, the molecular, the sort of mouse up to the human level. I think that's potentially very, very exciting.
Thank you, Tim. I'm going to wrap up now because of time, but I'd like to thank Valentina and Tim again for really inspiring talks. You can see parallels between the talks as well, which I found really interesting. If you haven't joined the Demon Network, then do go to demondementia.com, and it's free to join. And we are going to be having these seminars on a monthly basis, and every other month it will be jointly with the DRI. So thank you again to the DRI and to everyone who's has joined us. I think it's put us off to a great start. Thanks very much, everyone. Bye.
Thank you. Bye.
Bye.